Okay, so today we're going to continue discussing mapping with a focus on the kinds of calculations you have to do to construct your maps and to do probabilities associated with all that. Okay, so we already know actually how to draw genetic maps based on 2-gene recombination. And now what we're going to look at is different types of multiple crossovers, specifically double crossovers. And we're going to be looking at 3-gene segregation patterns and looking at 3-factor cross data. We're going to learn how to calculate gamete types given a refrequency map and calculate the coefficient of coincidence and interference. Now this sounds a little bit intimidating, but all the calculations are really very simple, and they're all the same once you get used to them. Okay, so reminder, if we have, of course, independently assorting genes, we will produce all 4 gamete types or all 8 gamete types in equal frequency because of independent assortment. Okay, now if you have linked genes where they're part of the same DNA molecule, you can't get those other gamete types unless you have crossovers between the genes. So that's represented here. We have the parentals, which are big A, big B, and little a, little b, and you put those together to make a heterozygote. We know that those 2 genes are linked, so we have to have a special way of representing this fact. And here we show it as big A, big B over little a, little b, with a horizontal line indicating the chromosome. So that horizontal line tells you that the genes are linked. It tells you they're actually linked in that particular configuration, a fact that we'll get to a little bit later. Now you can draw a crossover fairly easily if you put all 4 chromatids together and then do your exchange, and you can see where you can generate parentals and recombinants, 2 of each, as a result of this single crossover. And that's actually what's happening in these things, but it's actually a lot easier to represent that more simply the way I have it on the lower left part of the picture, where we make a little squiggle that goes from the big A to the little b, and from the little a to the big B. You're considering only those chromatins which are actually involved in the exchange. And it's the same idea, but it's just a simpler representation of the way that you go about getting recombinants. Okay, so intuitively, again, we have to realize that closely linked genes will have very little recombination between them, and I've represented that as a big A, big B, written very close together. You do get all 4 gamete types, but the recombinant types will be relatively few because the genes are closely linked on the same chromosome. Okay, by contrast, if the 2 genes are relatively far apart, there will be this big space between them, more opportunity for recombination, and you will have commensurately more recombinants forming, and that will be a measure of the map distance between. Now, I want to point this out now. If genes are far enough apart on the same chromosome, they will assort independently as measured in a cross, and it will take other crosses to know that they're actually part of the same chromosome. So chromosomes can be hundreds of map units long, and so the genes that are far apart will appear in a cross to assort independently. They will assort independently, but they're actually still physically linked to one another, which can complicate issues sometimes. Okay, so here's a representative map. This is from Drosophila melanogaster, and it just illustrates the idea that you can have your genes spread along the chromosome, and that chromosomes can have more than 50 map units on them. This chromosome, for example, has 107 map units, and this one has 106, meaning that the genes that are located far apart in that chromosome will appear to sort independently in a cross, even though they are physically linked to each other. So you have to do multiple crosses along the length of the chromosome to actually put the whole map together in this case. Okay. So in accord with that, map distances are not really additive, for the simple reason that you can have more and more exchanges between, more and more crossovers between, but you can never measure more than 50% recombination in a cross. Because remember, independent assortment gives you 2 recombinants and 2 parentals, which if you add that up is 50% recombinant. So that's the maximum you can measure in a cross. So even at relatively shorter distances, there is a departure from additivity as you get further, as the genes get further and further away. Okay, so genes that are far apart on the same chromosome will experience multiple crossovers between them, with the result that recombinants will equal parentals. And so you end up randomizing your chromatids so that genes on the same chromosome do not show linkage even though they are physically part of the same DNA molecule. So we're usually undercounting crossovers. So this is a little map, and we're going to save this one for later. It's a little way of putting together multiple crosses to give you map units. And this one actually is additive. But give it a try, and we'll go over it next time. All right, so here's my answer that I put the pieces together. We're mapping genes A, B, A, D, B, and C by pairwise crosses using just pairs of them and measuring the map distances between them. And our little puzzle is just fitting them together in such a way that you get consistent map distances across this section of the genome. Okay, so showing that crossovers will randomize, we need to look at 2-strand double crossovers, 3-strand double crossovers, and 4-strand double crossovers, just to see their outcomes. Okay, so we can draw a single crossover here, where we have the two parentals, well, the two homologs are aligned for meiosis, and we're showing both chromatids on both chromosomes. And the little x denotes a crossover, a breakage and reunion exchange, where the little a is now going to be connected to the big B, and the reciprocal, big A, is going to be connected to the little b. So those are the recombinant progeny that arise from a single crossover between A and B. Well, one type of double crossover that can occur is a 2-strand double crossover, where the second crossover essentially undoes the effect of the first one. So here we have the top chromatid is not involved in the exchange. It will be parental. The bottom chromatid is also not involved. It's also going to be parental. But here we're going to look at the two chromatids that are involved. So here we have our big A. We're going to have a crossover to the other chromatid, and then we're going to move along the chromatid, and then we're going to have another crossover between A and B. So the effect of that second crossover is to undo the effects of the first one, because that's going to give us a parental, and the other one's going to give us, its reciprocal is also a parental. So it's going to look like we had no crossover, where in fact we had two crossovers. You can also have 3-strand double crossovers, in which you end up with 2 parentals and 2 recombinants. So what you're seeing is you observe the big A, little b, which is a recombinant, the big A, big B, which is a parental, little a, big B, which is a recombinant, and little a, little b, which is a parental. So the result is 2 recombinants and 2 parentals, which looks like one recombination event, where in fact you've had 2 recombination events, but you can't tell because of the way the genes are arranged and the distance between them. Okay, so finally we can look at a 4-strand double crossover that's on the bottom of this slide. Okay, and here we have, actually, you get 4, all recombinant. We have big A, little b, big A, little b, little a, big B, and little a, big B, following those chromatids along. So in that case where all 4 strands are involved in the double crossover, the recombinant, you get equal numbers. They're all recombinant progeny. Okay, so now we're going to consider calculating map distances from 3-factor cross data, which gives more information than simply 2 genes at once. So we're going to consider our heterozygote, which is big A, big B, little b, big C, little c, and we're going to cross it times a triple homozygous recessive in order to be able to assess the gametes that came from the heterozygote. Because we're crossing times a recessive, we can look at the progeny phenotype and infer what gametes produced it from the heterozygous other parent. Okay. So we're going to start with an arrangement that we're told, where we have big A, big B, big C on one homolog, little a, little b, little c on the other homolog. So from here, we know that the big A, big B, big C is a parental. Little a, little b, little c is also the parental. Okay, and we know we can predict what progeny will occur as a result of crossovers between A and B and between B and C, and double crossovers in which 2 recombination events, one between A and B and the other between B and C. Okay, so the progeny types from this heterozygote and where they come from. And this is important to understand. So if we have our original parental, which is big A, big B, big C, little a, little b, little c, and there's no crossover, the progeny will be of the parental phenotype when crossed times a homozygous recessive. So those are big A, big B, and big C, little a, little b, and little c. But we can generate recombinant types, not by independent assortment, but by crossing over. And if we have a crossover between A and B, we get a big A, little b, little c, a little a, big B, big C. So those are reciprocals of each other, and they represent a single crossover between genes A and B. And they will be less than the parentals because it takes a crossover to form them. And the crossover, of course, depends on the distance between the two. The other single crossover is between B and C. Here we have big A, big B, little c, little a, little b, and big C. So those are also, of course, reciprocals of each other, and they give the other two classes of recombinant progeny that form as a result of that single crossover. Okay, so the last two of the 8 gamete types are the double crossover progeny, and they will be, of course, less frequent than either of the single crossover types, and less frequent even more from the parentals. And so those progeny types are big A, little b, big C, little a, big B, and little c. And so those will be the rarest of the 8, and we'll be able to pick those out of a pile of numbers based on this fact. Okay, so reiterating, there are 8 gamete types, 8 progeny types, the 2 parentals, which have no crossover that you can measure, single crossovers between A and B or between B and C, and then the double crossover where you had a crossover between A and B and between B and C. Now these can't be predicted unless we know our map, because we don't know the distances ahead of time. And of course, again, reiterating, the progeny phenotype tells us which gamete was produced from the heterozygote parent to give us that particular progeny. So we're given, in this particular example, a pile of numbers which has been conveniently arranged for us. Okay, so they're already in pairs, the reciprocals. Reciprocals are similar to each other in number. And we've already been told the gene order. We know that it's gene order ABC. And those 2 reciprocals are 45 and 55. The B and C recombinants are 68 and 75, because we know what those were. And the relatively infrequent ones are the double crossovers, and those are 8 and 12. So if we have this all together at once, we know what's what. It's a simple matter to calculate the map distances and make a map. And these numbers conveniently add up to 1,000. Okay, so here's our map. From A to B, we count up all the progeny that experienced a crossover here, and then divide by the total. So these are certainly the single crossovers between A and B. Those are the big A, little b, little c, which is 45, and the little a, big B, big C, 55. But it doesn't seem fair, but you have to. You have to also count the double crossovers, because they had a crossover between A and B also. They also had a second one, but we have to count them for the A-B distance. So these are the big A, little b, big C, which are 8, little a, big B, little c, which are 12. So calculating this map distance between A and B, we have our 45 plus 55 plus 8 plus 12 over 1,000. That's 120 over 1,000, which is 0.120, times 100 makes 12.0 map units. Okay, the B to C distance is calculated similarly. We take the single crossover progeny, which are 68 plus 75, and we have to count the double crossovers again, because they had a crossover here also. And that's 68 plus 75 plus 8 plus 12 is 163 over 1,000, which is 0.163, or 16.3 map units, or centimorgans. So if the information is already prearranged for you and reciprocals are identified, it's really pretty easy to calculate these distances. Okay, so here's our final map. A to B is 12 centimorgans, B to C is 16.3 centimorgans. Okay, so now we're going to do one on the handout where you have to find and pair the reciprocals first. So the way you do this is to look for the letters, but also look for numbers that are close to one another. The frequent ones are the parentals, the rare ones are the double crossovers, and the ones with intermediate numbers are the single crossover classes. So here's our pile of data. not in a nice organized form, but one that we can easily piece apart. We can see that big A, big B, big C is 300, and we know that it's a parental, and we know that its reciprocal is little a, little b, little c. Similarly, we can identify the single crossover products that match each other numerically and also having complementary alleles. So the big A, little b, little c is 100, the little a, big B, big C is 110. So those are single crossovers between A and B. The big A, big B, little c, and the little a, big B, big C is a reciprocal, and they're similar in number, as diagrammed. All right, so I've simply circled the reciprocals so that we know what's who, and then it's a simple matter of pairing them and doing the calculation to get the map units. So my answer is that A to B is 22.5 and B to C is 17.0 map units. So you should try this one and make sure you get the same answer. Okay, so now that we know how to make a map given progeny phenotypes, gamete genotypes, we can work the whole process in reverse by getting a map and then predicting what genotypes and phenotypes the progeny will get based on those distances. So we're going to use this example map, which is genes A to B, 10 map units, B to C, 15 map units. Now, for probabilities and prediction purposes, we don't use the map units, we use the frequencies. So for the first distance, it's 0.10 for 10 map units. For the second distance, it's 0.15 frequency. Okay, we're calling these region 1 and region 2. In order to actually make predictions, we have to know what the order and configuration of the alleles will be, and we're going to use big A, big B, big C on one homolog, little a, big B, little c on the other, linked in that order at those distances. Okay, so predicting gamete types or progeny phenotypes, the easiest one to start with to understand is the double crossover. So our progeny, big A, little b, big C, little a, big B, little c, those are the double crossover progeny. So they come from two crossovers, one in region 1 and one in region 2, between A and B, between B and C. So this is a simple probability. The probability of both events together is the product of the separate probabilities. So we have probability of 0.1, probability of 0.15. So we simply multiply those together to get probability of 0.015 as the product of the separate probabilities. So that's the probability of a double crossover occurring within this map region. Now, the slightly tricky part about this, that's just the probability of a double crossover, which actually has two progeny types. So the probability of each of those progeny types is half of that, which is 0.0075. So together they're 0.015, individually they're 0.0075. Okay, so the remaining probabilities are a little harder to see. The parentals will occur because there's no crossover in either region 1 or region 2. So remember, it's 10 map units and 15. So we have to say the non-crossover probability, well, either it crosses over there or it does not. So the non-crossover probability is 1 minus the crossover probability. So in this example, the probability of not crossing over between A and B is 1 minus 0.1, which is 0.9. And the probability of not crossing over between B and C is 1 minus 0.15, or 0.85, which multiplies out to be 0.765. Now again, that's for the two parentals together. For each one, it's half of that, which is 0.3825. Okay, so for the single crossover progeny, you have to know where they came from. The big A, little b, little c, and the little a, big B, big C. They come from a crossover between A and B, and not a crossover between B and C. The other single crossover is similar. The big A, big B, little c, little a, little b, big C, come from no crossover between A and B, but yes, a crossover between B and C. So for that pair, the big A, little b, little c, and little a, big B, big C pair, the calculation is there is a crossover between A and B, which is 0.1, and there is not a crossover between B and C, which is 1 minus that, and that multiplies out to be 0.0850. Individually, that's 0.0425. For the big A, big B, big C, and little a, little b, big C pair, that means we do not want to cross over between A and B, but we do want to cross over between B and C. So that's the probability is 0.9, 1 minus 0.1, times 0.15, which together is 0.135, and divide by 2 to get the individual ones is 0.0675. Right, so here is this all written out exactly the same with everything taken into account. Okay. So another way we can use this is just to look for individual gamete types or individual progeny rather than having to go through the whole mess. What fraction of progeny will have no crossovers in this region is no crossover here, 1 minus 0.05, no crossover here, 1 minus 0.3. Right? So that gives me 0.665. Okay, this example is just a little bit different because the arrangement of the alleles is different. In this instance, we have not all the dominants together and not all the recessives. We have them mixed up. So we have the big A, little b, little c on one homolog, and the little a, big B, big C on the other. So here the parentals are the big A, little b, little c, and the little a, big B, little c. So you have to take this arrangement or configuration into account when figuring out the probability of particular progeny. You have to know what your starting material is. So we're asked what the probability is of a little a, big B, little c gamete, and so we have to know where that would come from. So that comes from a non-crossover between A and B, because those are both one of the parentals from that, but there is a crossover between B and C. So we have to calculate that as a non-crossover between A and B, and yes crossover between B and C, times a half, because we're only looking for one of the progeny classes, not both of them. Okay, so that gives me 0.95 times 0.3 times a half, which equals 0.1425. Okay, finally, there's something called interference, which is calculated using another thing called a coefficient of coincidence. So interference is the idea where if you have a crossover, it inhibits the occurrence of other crossovers nearby. So that uses the map distance frequencies and is calculated using the number of crossovers that you actually observe in a cross divided by the expected number based on the map distance. So is that like confusing enough or what? Interference is calculated as I interference is 1 minus that ratio, which tells us whether there's any effect of one crossover on another one. So in our example, we're going to say that we have 9 double crossovers out of 1,000 progeny. So our observed frequency is 9 divided by 1,000, or 0.009. The expected frequency is the map distances multiplied. So the expected frequency of double crossovers is 0.05, which is to say the probability of crossover between A and B, and times 0.3 crossover between B and C. So 0.015 is the expected frequency of crossing over. So we calculate the coefficient of coincidence simply by taking the observed frequency, dividing it by the expected, and that gives us 0.6. The interference is 1 minus this. 1 minus this equals 0.4. Okay, so yes, there is interference because the observed is less than the expected. We observed 9 and expected 15. So we're going to say, yes, there is interference because there's fewer than we expected to get. If expected and observed are the same, the coefficient of coincidence is 1 and interference is 0. If there are no double crossovers, then interference is 1 or there's complete interference.